I'm Nate Martin, and today we'll be talking about CRISPR/Cas9 gene editing. Now, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which refers to the sequence of DNA that CRISPR interacts with. And we didn't really invent CRISPR, more so we hijacked CRISPR from bacterial immune systems. So just like there are viruses called phages that infect humans, there are also viral strains that target bacteria, which are very creatively named bacteriophages. Bacteriophages are actually being looked at as a possible solution to antibiotic-resistant bacteria, using gene editing like CRISPR to design custom viruses to fight bacterial infections. In essence, using the bacteria's defense system against it. But I digress. For bacteria, CRISPR works like memory T cells in the human immune system, allowing bacteria to remember a certain virus. If that virus strikes again, CRISPR produces RNA sequences that are inserted into the virus's DNA using an enzyme called CAS9, hence the CRISPR/Cas9. And this sort of biological cut and paste kills the virus. So people, in our endless ingenuity, figured out that we could leverage this. CRISPR was first discovered to be an adaptive immune system by Francisco Mojica at the University of Alicante in 2005. But it wasn't adapted and used as a gene editing tool until 2013, when Dr. Feng Zhang and his team at the Broad Institute. Here's how it do what it do. We can synthetically manufacture a guide RNA sequence, which functions like a blueprint, telling the Cas9 enzyme what it is to cut and paste. Cas9 is an enzyme, so it's just a protein that's structured for a particular function. If we imagine DNA, the fundamental building blocks of all life, as a Microsoft Word document, then Cas9 is essentially find and replace. Cas9 locates in the DNA the sequence that's been programmed into it, and it makes a cut on that sequence, removing it. Then a different, modified Cas9 with a template RNA sequence is sent to the site and sticks to whatever sequence it has been programmed with into the gap. Why is this useful, you may ask? As you may know, dear viewer, there are a great many diseases with primarily genetic components, such as cystic fibrosis, many varieties of cancer, and even the HIV infection. For example, here we have a stormtrooper. This stormtrooper has HIV. Poor stormtrooper. Luckily for him, modern medicine may have a solution. HIV reprograms the DNA of his immune cells. Let's call this block here HIV. To produce more HIV, which lies an insidious weight inside his cells. But with CRISPR/Cas9, as shown with this Lego model here, we can make a template of HIV's viral RNA, which we can then program into the cutter variety of Cas9, which will go onto the DNA, scanning for a particular sequence, and when it finds it, it makes a break, removing that segment of DNA, and it continues on. Now then, we can also use the replace sequence of Cas9 with a functional, non-infected sequence of RNA programmed into it to scan over, and when it finds the defect, it replaces it. So Stormtrooper here now has a healthy genome. This effect will cascade through his cells by the body's own cell replacement mechanisms, and within weeks, he will be cured. Happy Stormtrooper. With college coming up, I've been thinking about doing engineering, but with a medical spin, so I did a topic on biomedical engineering. As for CRISPR specifically, it's been getting a lot of negative press recently because of an unethical experiment. So I wanted to make this video to dispel some of the fears around that and inform people about how the technology works. I still think that despite its possible misuses, CRISPR is a very useful technology that should be explored further. Thank you for watching.